Welcome back, everyone. We're here to unpack some really crucial radiation biology concepts today. Stuff that's essential, not just for understanding how radiation works day to day, but also for tackling those tricky challenges like tumor radioresistance.
Especially hypoxia.
Right, especially hypoxia. And let's be honest, for getting through those board exams, too.
Absolutely. Think of this as a focused session, really diving into the chemical agents, the drugs designed to change how cells respond to radiation. We're going to hit two main groups.
Okay.
First, the ones that make cells more sensitive when the radiation hits the radiosensitizers.
hmm
And second, those drugs that are toxic, mainly just because cells are in a low oxygen environment, the bioreductive drugs.
Got it. Sensitizers and bioreductives. And understanding these really helps us see the limits of RT, maybe ways to push past them.
That's the idea.
So, okay, before we jump into the drugs themselves, maybe just a quick refresher. Why is hypoxia such a problem? We've touched on the oxygen effect before.
Right, the oxygen effect. It's
Yes.
fundamental, really. Basically says that cells without enough oxygen are much, much harder to kill with our standard low LET radiation, like photons.
How much harder?
We're talking about 2.5 to 3 times more resistant compared to cells that have plenty of oxygen.
Wow, 2 to 3 times.
Yeah.
That's huge. So the dose we give might kill off the oxygenated parts, okay, but it could leave behind this core of really resistant hypoxic cells.
Exactly. And those survivors can then just regrow the tumor. So the whole point of the agents we're talking about today is to try and, well, either close that sensitivity gap, make the hypoxic cells act more like oxygenated cells when we radiate,
Oh.
or just find a way to kill those resistant hypoxic cells selectively, whether or not radiation is happening at that exact moment.
Okay, so that really sets up our two main strategies for today. Agents that boost the radiation damage right when it happens,
Radiosensitizers. Yep.
and agents that kill cells specifically because they're hypoxic.
Right. Those are the bioreductive drugs, sometimes called hypoxic cytotoxins. They get activated by the low oxygen.
Okay, let's tackle the first group, the classic radiosensitizers that work right on the DNA. Halogenated pyrimidines, is that right? halogenated pyrimidines. Heard them called Trojan horses.
That's a pretty good analogy,
Oh.
actually. The idea is quite neat. These drugs look a lot like thymidine, one of the DNA bases.
Okay.
The main examples you'll see are 5-bromodioxyuridine,
BRH-MODOKCEU or BEDR, and 5-iododioxyuridine.
iododeoxyuridines, IUDR.
Yep. So cells that are dividing, they're making new DNA in the S phase, right?
Right.
If these drugs are around, the cell's machinery kind of gets fooled and grabs them instead of the normal thymidine.
Sneaky, so they get built right into the new DNA strand.
Exactly, they get incorporated. Now, thymidine normally has a little methyl group, but BRDU has bromine, IUDR has iodine, these big, bulky halogen atoms.
And that makes a difference. Oh, yeah. Putting those bigger atoms in there kind of destabilizes the DNA backbone, weakens it, makes the strand physically easier to break when radiation hits it.
Huh. Like using weaker bricks in a wall.
Pretty much.
Oh.
And it's probably not just physical weakness. Having that halogen there might also mess with the cell's ability to repair the radiation damage properly.
Okay.
Plus, the actual chemical radicals formed when radiation zaps these modified bases might be sort of different, harder for the cell to fix.
So weaker structure and potentially interfering with repair. More lethal damage overall for the same dose.
That's the net effect, yeah. If you look at cell survival curves, you often see the shoulder region get smaller or disappears. That means less repair of sublethal damage.
Okay, makes sense. But you mentioned something else crucial about these, especially for exams, they have a dual role. Because they only go in during S phase.
Absolute critical point. Because they only get incorporated during DNA synthesis, they become amazing markers for figuring out which cells are actively dividing, proliferating.
How does that work in practice?
Well, in the lab, you can feed cells BRDU or IUDR for a bit.
Yeah.
Then you use antibodies that stick only to the BRDU or IUDR within the DNA, tag those antibodies with something fluorescent.
And then you can see which cells lit up.
Exactly. Under a microscope, or using flow cytometry, you can literally count the cells that were in S phase while the drug was present. It's how we measure the labeling index, a fundamental measure of proliferation.
Right. But clinically, we don't really use these as sensitizers much anymore, do we? What were the problems?
Well, to get enough into the tumor DNA to make a real difference, you needed to give the drug for a long time over multiple cell cycles.
Okay.
So the whole idea relied on tumor cells dividing way faster than normal tissues, which isn't always the case. And then there was toxicity. BRDU in particular, cause bad photosensitivity, nasty skin rashes in sunlight.
Ouch.
IUDR didn't have that issue as much, but honestly, neither really became standard for sensitization. The therapeutic gain just wasn't consistently big enough to outweigh the hassles and side effects. Still vital lab tools, though.
Okay, so that's messing with the DNA directly. What about tackling hypoxia more head-on, making those low oxygen cells react better during radiation? That brings us to the hypoxic cell radiosensitizers like nitroimidazoles, nitroimidazoles.
Exactly. This approach targets the oxygen effect mechanism itself. These drugs are electron-affinic. They really like electrons.
Okay, electron-affinic. Let's recap the oxygen fixation idea. Radiation hits DNA, makes a reactive spot, a radical.
A DNA.radical, yep.
And hypoxia, that can often be repaired, right? Chemically fixed back?
Often, yes.
Yeah.
It's transient. But if oxygen is there, oxygen is great at grabbing electrons. It reacts super fast with that DNA radical, forms a peroxyl radical, DNA O dot, and that reaction fixes the damage, makes it permanent, harder to repair.
So oxygen basically makes the radiation damage stick.
That's a good way to put it. It locks it in. And the nitroimidazoles, they act like oxygen mimics in that critical chemical step, but specifically in hypoxic cells. When a nitroimidazole bumps into that radiation-induced DNA dot radical, and there's no oxygen around, it can react with the radical. It forms a DNA sensitizer adduct.
Ah, so instead of DNA oxygen, it's DNA nitroimidazole.
Essentially, yes.
Yeah.
It chemically fixes the damage in a way that's less repairable, mimicking what oxygen does. But critically, the drug has to be there in the cell at the moment of irradiation for this to work.
Just like oxygen would need to be. Got it. And these can diffuse into hypoxic areas.
Generally, yes. They're designed to be stable enough to get into those poorly oxygenated zones.
Okay, key examples. Mesanidazole.
Mesanidazole, MYSO-NIDazole, was the big early one. A 2-nitroimidazole, studied a lot. But the big problem was dose-limiting peripheral neuropathy, neuropia, nerve damage. It limited how much you could safely give.
Right. Then etanidazole.
Etanidazole, ETU-NY-duzole, another 2-nitro, designed to be less neurotoxic, made it more water-loving, hydrophilic, H2-hydrophilic, hoping it wouldn't get into the central nervous system as much.
And nimorazole. That name pops up in head and neck cancer.
Nimorazole. That's a 5-nitroimidazole. It's actually less electron-affinic, so maybe less potent as a sensitizer.
What?
But also less toxic. And this is really key. Nimorazole is pretty much the only one in this class that showed a consistent, statistically significant benefit when added to radiation in big randomized trials. Specifically for head and neck cancer, the Hansa studies from Denmark.
Okay, the Hansa studies, nimorazole, head and neck. Definitely flag that.
Yeah, it's not used everywhere, but it's the closest thing to a clinical success story for sensitization in this group.
Now, you hinted at another use for these nitroimidazoles, maybe even more important clinically today, not treatment, but
Yes.
seeing.
Imaging. This is huge. Using radioactively labeled nitroimidazoles as tracers for PET scans to visualize tumor hypoxia.
Like F-MISO.
Exactly. Fluorine-18 misonidazole, F-MISO. Or another one is FAZA, fluorine-18 fluorizomycin arabinoside, fluoroizomycin arabinoside.
How does that work?
Okay, so you inject the tracer, it goes everywhere. But only in hypoxic cells, there are enzymes that perform a bioreduction reaction on the nitroimidazole part.
Bioreduction. Hang on, didn't we say that was the other category of drugs?
Good catch. Yes, it's the same fundamental process. In low oxygen, the drug gets reduced. And in this case, the reduced form of the tracer becomes super reactive and sticks, covalently binds to stuff inside the cell, proteins, DNA, etc. It gets trapped.
But not in oxic cells.
Right. In oxic cells, oxygen jumps in, reoxidizes the intermediate before it can bind, and the tracer just washes back out.
So more hypoxia means more trap tracer.
Precisely. The PET scanner sees where the radioactive fluorine-18 is concentrated, and that gives you a map of the hypoxic regions in the tumor.
That seems incredibly useful. What do we do with that information?
Well, for one, we know hypoxic tumors often have a worse prognosis. They're tougher to treat. So an F-MISO scan can help predict response. And looking ahead, maybe we can use these maps to guide treatment, like ghost painting, giving a higher radiation dose just to those hypoxic spots, or choosing patients for hypoxia-targeting drugs. It's a powerful, non-invasive tool.
Okay, really interesting connection between the basic mechanism and a current clinical tool. So we've done DNA modification, we've done mimicking oxygen. What's the third strategy? The bioreductive drugs or hypoxic
Oh,
cytotoxins.
right. This is a totally different angle. The idea here is to have a drug that is mostly harmless when you give it.
A prodrug.
A prodrug, exactly.
Right.
But it gets converted into a potent cell killer only when oxygen is scarce. It requires enzymatic reduction inside the cell to become active.
And oxygen stops this activation.
Yes. Remember how oxygen loves electrons? In a normal oxygenated cell, if an enzyme starts reducing one of these prodrugs, oxygen is right there to grab the electron back, instantly reoxidizing the drug intermediate back to its harmless starting form. It's like a futile cycle: reduce, oxidize, nothing happens.
But take away the oxygen.
And that reoxidation escape route is gone. The reduced intermediate hangs around long enough to undergo further reactions, rearrangements, and that creates the highly toxic species. Often nasty free radicals where things that damage DNA.
So the killer form only gets generated where there's no oxygen to shut down the process. That's the selectivity. How do we measure how selective it is?
The key metric here is the hypoxic cytotoxicity ratio, HCR. You absolutely need to understand this one.
Okay, break down HCR for us.
It's a ratio. It's the concentration of drug you need to kill a certain fraction of oxic cells, divided by the concentration of the same drug you need to kill the same fraction of hypoxic cells.
Okay, so hypoxic conc for the same kill level. What does a high HCR mean then? Like say HCR of 50.
An HCR of 50 means it takes 50 times more drug to get the same kill in oxic cells compared to hypoxic cells. Or, putting it the other way, the drug is 50 times more potent under hypoxic conditions. You only need 1/50th the dose in hypoxia for the same effect. So yeah, higher HCR means better selectivity for killing hypoxic cells.
Got it. Higher is a better selectivity.
Yeah.
Examples.
The classic textbook example, based on lab data anyway, is tirapazamine, also known as SR4233. It's an organic N oxide, gets reduced under hypoxia to a really toxic free radical.
Tirapazamine. I remember hearing about that one. Huge HCRs in the lab, right?
Oh yeah, sometimes 20, 50, even over 100 in some tests. And it looked amazing. In preclinical models worked great with radiation, with chemo like cisplatin, seemed perfect for wiping out that resistant hypoxic fraction. But it didn't really pan out in the large clinical trials. It was due to systemic toxicity or just didn't boost cure rates enough across the board. So despite the incredible HCR, it's not standard treatment. Real shame, in a way.
Yeah, tough translation from bench to bedside sometimes. What about mitomycin C? MY to MY-syn SCE. We actually use that one.
Right, mitomycin C. It's an older drug, an anti-tumor antibiotic, works as an alkylating agent. It also needs bioreductive activation to do its DNA cross-linking job.
Does it have a high HCR?
More modest, usually quoted in the range of maybe 2 to 10, so less selective for hypoxia than tirapazamine was.
But still somewhat selective.
Yes. It does kill oxic cells too, but its activity definitely gets a boost in hypoxic conditions because of that activation pathway. And we do use it with radiation, famously in anal cancer protocols. It thought that its ability to hit hypoxic cells contributes to why that combination works so well there, even with a modest HCR.
Okay, so looking back, halogenated pyrimidines, hypoxic cell sensitizers, bioreductives, it really seems like getting these things to work well in patients has been the tough part.
It really has. You know, with the pyrimidines, it was toxicity, needing fast proliferation. With the nitroimidazoles like masonidazole, it was neurotoxicity. Nomorazole had some wins, but niche. Tirapazamine, amazing HCR, but failed phase 3. Mitomycin C, modest HCR, but found its place.
It just hammers home that whole therapeutic gain issue, doesn't it? You have to kill the tumor more than you hurt the normal tissues.
Exactly, that therapeutic window. It's often just too narrow with these systemic agents. Normal tissues have hypoxic pockets too, or different sensitivities. And finding that selective advantage in the complex human body is way harder than in a petri dish.
All right, great overview. Let's try and crystallize some clinical pearls here, things to really take away.
Okay, pearl number 1. Halogenated pyrimidines, BRDU, IUDR. Remember them for 2 reasons. 1, the concept of DNA-targeted sensitization by incorporation. 2, and maybe more relevant now, they are indispensable lab tools for measuring cell proliferation, that labeling index. And don't forget BRDU's historical photosensitivity issue. Good one.
Yeah.
Okay, pearl number 2, hypoxia. Still a huge problem in Radiosensitizers. Direct sensitizers like narintonzole have had limited specific success, but the imaging side, using things like F18, misonidazole, PET, that's a really valuable clinical tool today for prognosis and potentially guiding future treatments, connects the basic science right to the clinic.
Excellent. And pearl 3, bioreductive drugs. They exploit hypoxia for selective killing. The key concept is the HCR hypoxic cytotoxicity ratio. Remember how it's calculated? Hypoxic conc for same kill. High HCR equals high selectivity. Think tirapazamine, high HCR, clinical disappointment. Think mitomycin C, modest HCR, but has found clinical niches like anal cancer.
Perfect. Ready for a quick board blitz? Test ourselves?
Let's do it.
Question 1. A patient gets BRDU before radiation. What's its main mechanism as a radiosensitizer? A. Scavenges free radicals. B. Inhibits DNA repair enzymes. C. Gets into DNA instead of thymidine, making DNA weaker. D. Accumulates in hypoxic cells, acts like oxygen.
Okay, it's got to be C. BRDU as a thymidine analog is incorporated, weakens the DNA. Correct. Question 2. Misonidazole and its relatives, like F18-meso, good for sensitizing and imaging hypoxia. What's the main reason for their sensitizing action in hypoxic cells? A. Directly cause double-strand breaks. B. Inhibit mitochondria. C. Act like oxygen, fix DNA radicals. D. Increase blood flow.
That's the oxygen mimic idea. They are electron-affinic, react with the DNA radical when oxygen isn't there, fixing the damage. So C.
C is correct. Question 3. A bioreductive drug has an HCR of 50. What does that mean? A. 50x more toxic to oxic cells. B. Need 50x more radiation with the drug for hypoxic QS. C. Enhances oxygen effect by 50x. D. 50x more potent at killing hypoxic cells than oxic cells.
Right, HCR is oxic over hypoxic concentration. So HCR of 50 means it's 50 times more potent in hypoxia. Need less drug. So D.
Correct. D. Last one. Which of these is a bioreductive hypoxic cytotoxin gets turned into a toxic radical mainly when oxygen is low? A. Amifostine, MIFOStine. B. IUDRC, misonidazole. D. Tirapazamine.
Okay, amifostine is a protector. IUDR incorporates into DNA. Misonidazole mimics oxygen. Tirapazamine is the classic bioreductive prodrug activated by hypoxia. So D.
D is correct. Nicely done. All right. Whew. Trying to keep all these mechanisms and drugs like misonidazole, namorazole, tirapazamine straight. It feels a bit like trying to recall every single step of glycolysis right after you walk out of the biochem final.
Yeah, I hear you. It's a lot. But thinking about the core mechanism helps group them. Is it going into DNA? Is it mimicking oxygen? Or is it only getting activated by hypoxia? Focusing on those categories and the key examples, plus why they did or didn't make it clinically, that's usually the way.
Okay, so quick wrap up. We looked at different ways drugs try to beat radioresistance, mainly hypoxia.
Yep. Halogenated pyrimidines like BRDU, IUDR get into DNA, weaken it. Great for lab proliferation studies, limited clinical sensitization.
Hypoxic cell radiosensitizers, the nitroimidazoles. Meso, etonidazole hit toxicity walls. Namorazole had some specific H&N success, but F18-meso for PD imaging is a big clinical contribution now.
And the bioreductive's hypoxic cytotoxins need low oxygen for activation. Tirapazamine, mitomycin C. Remember HCR for selectivity? Tirapazamine had high HCR, failed clinically. Mitomycin C, lower HCR, but used in some settings.
And it all comes back to that challenge of therapeutic gain, hitting the tumor hard without hurting the patient too much.
Exactly. Now, we've mostly talked about making tumors more sensitive or killing the resistant bits today, but there's the flip side. What about protecting the normal tissues?
Ah, the other half of the equation. Good point. Next time, let's tackle that. We'll get into radioprotectors, like amifostine, and then explore using heat hyperthermia as another way to modify radiation effects.
Sounds good. And remember, you can find practice oral boards and other review materials over at radonsmartlearn.com. That's RADONC smartlearn.com. Make sure to subscribe to Radon Smart Review for our next session. Thanks for joining us.
And here's a final thought. If those hypoxic cells are so resistant, how fast do we really need to eliminate them during treatment? Does waiting give them time to reoxygenate, or just time to cause trouble? Something to ponder. See you next time.